Well, thank you so much, Bruce, for inviting me. I have to say, Carl June's talk was just a triumph, and I feel, like in my talk, we just started this kind of work, and there's a lot to be discovered, so it's more of a discovery platform than that kind of huge lifelong success that he described. So basically, I'm going to talk about exosomes as potential biomarkers for brain tumors. They can be potential biomarkers for any type of cancer, and probably for other types of cancer, they might be easier to use, but for brain tumors, we really have a need for biomarkers, so it's kind of the driving force. So my focus for a number of years has been glioblastoma. It's basically a virtually untreatable, highly malignant tumor of the brain. There are, you can usually keep the patient alive for a year or so with, surgical resection, radiation, chemotherapy. But really, there's not much you can do for these patients. And beyond that, because the tumor's in the brain, to even find out what mutations it has, you have to biopsy it. And then after the patient's undergoing treatment, there's usually a necrosis in the brain region. It's very hard to even know when the tumor's regrowing. It's very hard to monitor responses to drug therapy because basically you can't really track the tumor that well. So as I said, there's a big need for biomarker therapy in this particular type of tumor. So we actually came upon extracellular vesicles by accident, but in retrospect, they're actually a very interesting vehicle. The NIH Commons has actually targeted them now for an RFA to find out more about them. Exosomes is one of the terms used to describe these types of vesicles, but actually there's a whole number of different types of vesicles. So cells release a lot of membrane-bound vesicles, and they've been called a lot of different names depending on what cells release them. Things like microparticles, shed microvesicles, exosomes, retroviral-like particles, et cetera. At this point, I think there's pretty much agreement that we collectively, as a scientific community, do not know how to separate all these types of vesicles and do not know what their properties are, and that's what we need to tackle in the future. They are released by all cells, so they're in all bodily fluids here. And people believe that they're primarily used as a form of intracellular communication so that one cell can transfer charged nucleic acids like DNA or typically RNA from one cell to another. Also, if you have proteins, there's, say, cytoplasmic proteins that aren't secreted, or for instance, transcription factors, they can be sent over in these vesicles. But in addition, the cell uses them as a means of getting rid of toxic substances. For instance, a lot of tumors will use these vesicles to get rid of drugs. So they have kind of a number of different functions, and as I said, there's a lot of different types of particles, so sorting out which types of particles are doing what is something for the future. The contents are very large in a typical extracellular vesicle. So there's a limiting membrane, and there's all kinds of receptor and adhesion molecules in the membrane, MHC class proteins, death factors, and then inside you've got a lot of other proteins, including, like I said, transcription factors, and a lot of species of RNA. And that's really what our focus has been. And that was kind of even though if you go back historically, people going back to the 1970s, thought there might be RNA in these vesicles. Other people doubted them. It wasn't until the technology and nucleic acids reached a point where people could be very firm about the fact that, yes, they do contain a lot of different kinds of RNA, including messenger RNAs and microRNAs. I might say, though, that these aren't the only carriers in the bodily fluids of microRNAs and other RNAs. People now know that there are ribonuclear protein particles, there's HDA, high-density lipoprotein particles that will hold RNA molecules and protect them from being degraded. So in a recent estimates that I went from from a meeting I went to for microRNAs, they say probably in the circulation, about half of them are probably in these vesicles, these membrane-bound vesicles, and the other half are probably in these protein-lipid complexes that protect them from RNases. So as I said, we came upon our interest in these by accident. This is a tumor cell from a GBM patient from Mass General Hospital that we put into culture. I always remember it because Johan Skag, who really initiated this work in our laboratory, he was a PhD. I said, oh, you can't get clearance to go into the operating room. Bob Carter, the neurosurgeon, said no problem. But then he got the cells, and he couldn't ride on the shuttle bus, so he had to ride his bike from, basically from the operating room to the lab to put these in culture. And right away, we saw that these looked very strange compared to all the other types of glioma cells that we'd been growing continuously in culture, because they were just covered with vesicles. And we went on to characterize those. And you can see they have different sizes. They range in size. Most of them are 50 to 200 nanometers in diameter, but they can be very large. And some cancer cells do release these very large vesicles, and they've been called oncosomes. And we also isolated the RNA from them. And you can see this is just a marker, but there's a peak of RNAs in the 200 nucleotide range. They spread out to maybe 4 kb, and these two little peaks here are the ribosomal peaks. So in cells, those ribosomal peaks would be huge, but in the vesicles, there's very little of the ribosomal RNA. So these are just characterizing these vesicles released by tumor cells in culture, and these are actually medulloblastoma cells compared to fibroblasts. So one of the things that you see is that the size range is pretty similar to the vesicles, but the tumor cells release more vesicles. Here, this is about 20,000 vesicles released per cell over a 48-hour period, compared to maybe about 5,000 for normal cells that we looked at. There's also a lot more RNA in the vesicles, really a lot more compared to what you would find in vesicles, at least from the normal cells that we looked at. So here, looking at RNA, you can see a lot more RNA in the tumor vesicles. And the other interesting thing was, actually, this is the RNA here. We also found a lot of DNA in these particular vesicles. Actually, I think that's because these medulloblastoma cells were amplified for c-Myc, and there's actually a lot of c-Myc DNA in the vesicles, but I'm not going to go into that study. That's published in this paper here. So just to give a little background, this is what interests a lot of people about these vesicles, that as I said, they may serve as a means of intracellular communication. They contain surface receptors from what we call the donor cell. We think of that, in our case, as the tumor cell. They can be released by budding off the cell surface. They can be released from multivesicular bodies. The outer membrane also is like the outer membrane of the cell, so it presents the receptors. Those receptors, people have shown, can just be transferred to other recipient cells. You might think of that as a stromal cell in the tumor cell environment. So the vesicles can fuse with the recipient cell. They can be taken up by endocytosis, and they bring in a lot of information. I mean, when you just take, let's say, as we did endothelial cells in culture, and you put on these extracellular vesicles from brain tumor cells, they practically change in front of your eyes, the phenotype. But it's because there's so much information in terms of receptors and ligands and transcription factors and RNAs, are just very potent informasomes, I guess you might call them. And the big interest, at least at NIH now and in other camps, is in the actual RNA content. So in addition to containing some messenger RNAs, they contain a lot of non-coding RNAs. Those are microRNAs, but they also include this huge emerging class of non-coding RNAs that we think is involved in regulating transcription, here showing epigenetic modification, as well as inhibition of translation, splicing, et cetera. So there's a lot of information probably in that non-coding RNA component. This just shows you the avid uptake. So if you put here tumor cells labeled with a PKH fluorescent dye, you can label them in other ways also, in a chamber, and here are your recipient cells in the lower chamber. you can just watch within 48 hours, these cells will just gobble up these vesicles and become fluorescent also. So that's the other thing about the vesicles. Not only are they released in abundance, but they're very avidly taken up. There probably is some selectivity in the uptake process, but the determinants of that have not been well defined. This is just an experiment that was done. We did in the early days, showing that you could incorporate a messenger RNA into these vesicles, and in this case, encoding a luciferase molecule that isn't normally present. And when you treat cells in culture, these were endothelial cells, brain endothelial cells, you can see that the protein is actually synthesized in the recipient cells, and that's been shown by other people. So although most of the RNA in these vesicles is not very large, and you don't think of the message as being the most predominant RNA in there, especially if you overexpress it in the donor cell, you can get quite a lot into the vesicles, and then they can be translated in the recipient cells. So in terms of tumors, there's a lot of interest in how these vesicles change the environment, the stroma of the tumor. And just here, at some point, there's the mutations going on in the tumors. They're different in different people. These tumor cells release a lot of vesicles, probably a lot of different vesicle types. And people have shown that they play a role in immune suppression of tumors. They play a role in just opening up through protease action, but MMPs, the extracellular space, so the tumor cells can invade more quickly. Other people have shown for peripheral tumors that they're very involved in metastases. They set up zones for new metastases, and they stimulate angiogenesis. And for brain tumors, what we wondered from the beginning was, could they get into the bloodstream, or let's say into the CSF, and give us a marker of the genotype of the tumor. So this, I'm just going to put this all on. This is work that was done a while ago. It still isn't published because it still needs to be repeated, and this amount of ribosomal RNA is way overestimated, but this is just doing deep sequencing of RNA from some vesicles from these GBM cells. And just to say, even though there are coding sequences there, messenger RNAs, those may be fragments, those probably most cases they are fragments of messages, there is a lot of non-coding RNA. And you'll see this again and again. And this is the zone that we really don't know a lot about yet, but we think that may actually have more information for us than the messenger RNA analysis, for example. So here, just showing that if you analyze the messenger RNAs in these extracellular vesicles from the GBMs, and you look at the types of messages they are, a lot of them are involved in processes that would support tumor growth, angiogenesis, cell proliferation, usually immune response. And you'll see also that this is an indication of how much higher they can be. So some of those messages, or at least the fragments of them that we're quantitating, are up to 1,000-fold higher in the vesicles as compared to the cells. So it's if the tumor cells are actually placing certain messages into these vesicles with the intent of changing the environment around them in favor of tumor growth. At least that's the theory. And then I just wanted to, so as I mentioned before, there's most of what's in those extracellular vesicles is non-coding RNA. Most of that is not microRNA. It is, all these weird RNAs that people are starting to, so there is some microRNA, not a lot. There's some, what we call just more like antisense siRNA, maybe long non-codings that can do epigenetic regulation of methylation. These are also epigenetically acting to modify transcription off the genome. So, and then I'll point out here the retrotransposons. About half of our genome is retrotransposons, and tumor cells typically elevate the level of expression of these, and so they become quite abundant in the tumor microvesicles. So our questions were in terms of GBMs. Are these extracellular vesicles released into the bloodstream in the CSF? Do they have a unique profile? If we compare, let's say, patients compared to controls, are there certain messenger RNAs and microRNAs that are high in the tumors that would be high then in these extracellular fractions in serum and CSF? Can we pick out mutant messages in these vesicles in tumor patients? And how can we increase sensitivity? So the real kind of wall we come up against is that all cells release extracellular vesicles, essentially all normal cells. So the blood is filled with vesicles. They're filled with vesicles that are produced by endothelial cells, platelets, neutrophils. Actually, the most abundant are the ones that are released by the platelets. So we have a sea of vesicles, certainly in the serum, and now we're adding in a little dollop of these tumor microvesicles, and we're wondering, can we actually pick up mutations in that small fraction? So there's a couple of things that are probably in our favor. One is, if you just look at the number, these would be extracellular vesicles per mil in controls versus, let's say, GBM patients, you do have a lot more. Not a lot more, but some more in the patients. Hopefully, some of these are tumor microvesicles, but they can also be vesicles released by normal cells in response to the tumor. But having more seems good, and you hope that some more of those are due to the tumor microvesicles. This is a comparison just of serum and CSF. Obviously, with brain tumor patients, the CSF is going to be a lot closer to the tumor. We think it's, in some ways, it's going to give us a better picture of, it's harder to get, but it's going to give us a better picture of the mutations in the tumor. And this is just to show that the vesicles in serum or CSF are about the same size as in serum, but there's a lot less of them. But of course, we think a much higher fraction of them will be derived from the tumor in the case of a brain tumor. So this is a study we did where we just took GBM patients and controls and this was actually done by microanalysis of messenger RNAs and then analyzing the different relative amounts of different messages. And you can see here for 206 genes, if you do unsupervised clustering, you get distinct pictures of the messenger RNAs in the vesicles for the brain tumor patients compared to the controls. And actually, when we did melanoma patients in their serum, that looked even different. So you could almost say, well, you could tell just from looking at these set of messenger RNAs in microvesicles almost whether somebody had a tumor or not, and if so, what kind of tumor they had. But the interesting thing about this was that the messages that were the most different didn't seem to come from the tumor cells. They actually seemed to come as a response of the body to the presence of the tumor. So they tell us that the body's responding to a tumor in a sense, but they don't tell us anything about, in this case, the genetic content of the tumor. The other issue here is that if you look, remember I told you that the vesicles from the brain tumor patients do have more RNA, but if you actually look at messenger RNA, they actually have less of that. What they have here, again, on a bioanalyzer, this is the size, is this huge amount of these actually small non-coding RNAs, even less than, well, certainly less than 200 nucleotides. So that's what they're really rich in, and that's, we don't know a lot about non-coding RNAs, so that's an area that's going to need, in order for us to get information out of this and what it can tell us about the state of a tumor is going to require a lot more research. We did show, for example, just looking now here at serum EVs, and these are CT values, quantitation, so it's the inverse in terms of how high it is. If we look at tenascin C, which is a protein that's very highly expressed in GBM tumors, you can see there's more of that message in the circulating vesicles from the patients as compared to the controls by about 8-fold. If we look at a microRNA, miR21, that again is very high in patients, or very high in tumors compared to brain tissue, normal brain tissue, or other cell types. Again, big difference. And this was a set of, I think it was maybe 6 patients and 6 controls. So you can see these quantitative differences of things you expect to be elevated in the tumors compared to the normal cells, but it doesn't really tell you that it came from the tumor. It just, there could be a response of the body again to the presence of the tumor. Other things that we found a lot of were retrotransposon elements. As I said, in general, tumor genomes are hypomethylated, so they tend to express a lot of their repeat elements, including L1s and ALUs. And we did see those at high levels in GBM cells, both in terms of the quantity and then also in terms of enrichment in the vesicles compared to the cells. So in some cases, for some of these sequences, they were over 100-fold enriched in the vesicles versus the cells. Again, telling us, in this case, that there's a tumor present in the patient. This is looking at, these are human endogenous retrovirus RNA. These get turned on very highly in subtumors. And for instance, in breast carcinomas, they're turned on so high, and there's so many of them. Even though none of the HERVs is a replication-competent retrovirus, they can make, some make a perfectly good M proteins, some make perfectly good GAG proteins, some make full-length RNAs, et cetera. They can actually make particles that aren't infectious, but you can actually see them in the bloodstream, and you can see them in some of these tumors like breast cancers. Even in GBMs here, we see very high levels of these HERV-K, in this case HERV-K RNA. That's one of the most recently introduced into the human genome, meaning millions and millions of years ago. And although we haven't seen particles per se, in fact, the way people isolate vesicles from serum or anything else, you can't distinguish, let's say, an exosome from a retrovirus particle. You're just collecting them all. So we probably are collecting these particles. And just interestingly here, if you take, these are HUVEC cells incubated with these extracellular vesicles from medullobastoma cells, you can see that they take them up and they even retain the sequences for some time. Whether they can actually then integrate into the genome of the recipient cell, that's another area of very active investigation. So, as I said, you definitely see changes or distinctive markers in terms of high non-coding, small non-coding RNAs, HERVs, L1s, ALUs, and these extracellular vesicles released from the tumor cells. And some of them, especially like I'm sure some of the non-codings and some of the HERVs are going to turn out to be unique markers, but right now they haven't been that well defined. So we wanted to look at actual mutations that we know are present in some GBMs, and ask whether we could pick up that mutation in the serum or the CSF from the patients. So this is the EGFR. It's either called a mutant or a variant because it can arise either through mutation or through a splice variant. So this is actually, all the classic GBMs are amplified for EGFR, and this mutant protein is present in actually a large percentage, and that's why it's used for targeting. And I think Carl June was saying that's one of the targets they want to go after in GBM patients with their T-cell strategy. This is just showing that you can't, taking exosomes or extracellular vesicles from serum, you can quantitate levels of the wild type and EGFR. These are in patients, different patients that were positive for the mutation. You can pick it up in a number of these patients, and it's present in about maybe 1% to 5% of copies of the EGFR. So it's quite robust. This is the most robust marker we found. So just in recent studies, just looking at mutation status. So here, in the cases where the tumors were positive for EGFRV3, we picked up about 50% of them in the serum. And we tend not to pick it up at all if the tumor is negative, but 50% isn't great. And we also looked at CSF now. We have fewer samples, but there it's very good at picking up the fact the mutation is there. There are some false positives, but at least in the case of one of those false positives, it did have a mutation, a different mutation in the EGFR receptor gene. So this is probably the most promising assay that's coming along and will go into clinical trials. You can say, well, typically you're going to go in and take the tumor out and you're going to see that the person has an EGFR-V3 tumor, but the question really comes in when you're treating the patient and you want to track this tumor over time. You know, can you take the serum sample is not going to be that reliable. The CSF sample will probably be very reliable, but then you're like, neurosurgeons kind of fall into two camps. Half of them say, we don't want to take CSF from the patient. The other half says, oh, I can just put in a Meyer reservoir and I can just, take periodic samples and we can see what's happening. So I think, though, given the high specificity and sensitivity, it's definitely going to be something that will be looked at in terms of clinical efficacy. Another mutation that we've gone after is in IDH1, which is found in, this is the most common mutation, and that's found in a lot of low-grade or lower-grade gliomas, grade 2 and 3. Oops. Why isn't it going forward? The light works, goes back. Oh, there it goes, okay. So we started an initial study. Our initial work was done with Linda Liao, who's a neurosurgeon at UCLA. And we took, in this case, you'll see with CSF samples, she happened to collect CSF samples routinely on her patients. Some neurosurgeons do. Here in our initial study, 3 out of 4, we could pick up the mutation. Oh, sorry. Thank you. 3 out of 4, we could pick up the mutation in the positive tumors. These are the positive tumors here with the mutation, and here are the positives in the serum. And the one that was negative was the smallest tumor, so we were quite encouraged by that initially, but we've gotten more samples, and we're getting a little less encouraged. For the EGFR-V3, we can just do a qRT-PCR because it's a very big mutation. Here, it's a single nucleotide change, so we used a type of digital sequencing called beaming, which is a little more sensitive. We haven't been able to pick it up in serum at all. In CSF samples from positive tumors, we can pick it up in about half of them. So I think we're hoping at this point just to switch to deep sequencing. I think there was a paper out on that for, at least for EGFR-V3, and hopefully we can increase our sensitivity a lot more with deep sequencing. So I'm just trying to look at my time. How much time do I have left? About maybe. Okay, 5 minutes. Okay. So just to say, we've set up a consortium of 17 different institutions that are now all collecting CSF and serum samples under SOP conditions, so they're all the same. We can get, we're getting hundreds of samples. We have 600 GBM patients to date that have been collected. So this is going to give us the power to actually see how valid some of these assays are. Bill Butler here set up a great system where we have a virtual repository and we can literally track every vial in each place in time and how much has been used and where it is, and that's really been a huge help. We're looking at a couple of ways to increase biomarker sensitivity because, as I said, these few tumor microvesicles are in a sea of normal microvesicles. So we need to, if we could pull them out, we could get a higher sensitivity. We've worked actually with the group in the Cancer Center on this, using their technologies for isolating cancer tumor cells and trying to apply them now to microvesicles with antibody capture. I am just going to say that we're, based on their work, where they showed that you can capture human GBM cells anyway with an antibody for EGFR. We've used the same kind of strategy to try to capture with EGFR antibodies. We actually have a very involved strategy where we have mouse GBM cells, but they express human EGFR and EGFRv3, and we mix them with serum samples. We put them through a device that's loaded either with antibodies for EGFR or for just IgG, and then we analyze our capture. And if we're capturing these tumor cells here, we should see this high EGFRv3, high EGFR human, and low human, for instance, GAPDH, which is coming from the vesicles. And we basically, then after we've captured, we look at what we put in, we look at what we capture, we look at what we get out, we isolate the RNA, we do the qRT-PCR to quantitate it. And so far, we are starting to get good capture. This is the input. This is looking RNA now, this capture of this EGFRv3 message. Very little with the IgG, but the real problem is that we're losing a lot. So here, for instance, if you look at the input, in absolute copy numbers of this EGFRv3, we're still losing at least half of it. So I think there's, Shannon Stott is working with us on, tweaking this so that we can capture more. And I'm not sure I'm going to have time to go through this method, but suffice it to say that Ralph Weissleder has a diagnostic magnetic imaging device where he can, again, detect proteins on the surface of vesicles. That's a cell, but he's doing it on vesicles. And he uses an antibody capture of magnetic particles. So basically, your vesicles, if they're containing a particular protein, and this can be a protein on the surface or on the inside, will cause clustering, and you'll get a change in the T2-weighted signal. And this is highly sensitive for picking up proteins. If you compare it, for instance, to an ELISA, it's 1,000-fold more sensitive. And he can pick up EGFRv3 on these vesicles in serum, and he can pick up mutant IDH1. And that paper is coming out, I think, this month, finally. So we're looking at the RNA, he's looking at the protein, but I think there's a big push now to see what information we can gain from these vesicles in terms of mutations that are present in the tumor. So these vesicles do contain a representation of the transcriptome of the tumor. Highly sensitive nucleic acid techniques, as well as these new protein techniques, can allow you to pick up some of these mutations. We're hoping to increase sensitivity and to use this biomarker strategy to basically track patients during therapy and response. And this is the, whoops. This is the team. I'm not going to have a chance to go over everyone on this team, but my lab is listed at the top, and they've been very active in this approach, and we have a lot of collaborators in all fields of medicine and brain tumors. So thank you.
Thank you, Sandra. Are there questions from the audience? Jay.
Have you actually followed the exosomes in patients from diagnosis to therapy?
We haven't yet.
So the
but
dynamic changes?
Yeah, we haven't yet, but we've collected the samples to do it. So we have probably over, from MGH alone, probably over 30 patients that we have samples every 2 months, and we've just been holding those until we have the assays, validated, and then we'll process them.
Yeah. Joe.
Can you actually transfer DNA and get the DNA expressed from exosomes to a cell that captures the exosomes? In other words, can you transfer resistance, for example?
That's a very interesting question. We know that you can transfer DNA. For instance, if we have a tumor cell that's amplified for c-MYC, it will transfer that c-MYC DNA to the cells, but we don't know if it can be functional in the recipient cells at this point. But that is a
Sandra,
very interesting question.
you've spent a great deal of your effort on brain tumors. It would seem to me that that's a more difficult potential model because of the blood-brain barrier. Obviously, you can find some material of interest in the peripheral blood, but what about in large peripheral tumors, such as lung cancers? Could we sample exosomes and find evidence for mutations that are actionable?
Yeah, I mean, if the good thing is if you have a mutation that's, typical of that tumor, I would say for prostate cancer, it's even further along, and they actually get these vesicles from urine. And the other area where it's very active is melanoma right now, and that's also, so I think, anybody who has a tumor type and has a mutation and wants to join in on looking at this, we can help them, or, just...
I think that would be even more promising. I mean, I know why you're studying brain tumors. You're part of a neuro-oncology group. But it seemed to me that chances of success in actually finding potentially useful application in the short term would be with tumors in the systemic circulation.
Yeah, no, I agree with you. And we're very anxious to, like I said, either collaborate with or help anybody who wants to do that.
Right.